This is a quick little lecture about the endomembrane system. This goes along with Chapter 4, Section 4.5. And I just want to make sure as you're studying all your organelles and memorizing their structures and functions, that you understand that there is this system or series of organelles that's really very important in protein production. So we will revisit this in Chapter 10 when we talk about translation, but I wanted you to understand what this endomembrane system is. And endo in biology stands for inside or internal. So this is an internal membrane system. It consists of the nuclear envelope, the rough and smooth ER, the Golgi, and the Golgi is sometimes called the Golgi complex, the Golgi apparatus, all meaning the same thing. One thing I want you to make sure for the Golgi is that you always capitalize the G, because it's actually named after the scientist who discovered it. Okay, endoplasmic reticulum, lysosomes, everything else can be lowercase, and usually are, but the Golgi always has to be capitalized. Okay, so we have the nuclear envelope, the smooth and rough ER, the Golgi, the lysosomes, and what's not shown on here, peroxisomes. This is all part of this endomembrane system, which means the cell membranes are continuous in the sense that they can move or they can be transported from one of these organelles to another. So before we get into that, a couple things I want you to notice. One, the nuclear envelope and the endoplasmic reticulum are continuous, which means it is one system of membranes that's all connected. So if you look here, in purple is your nuclear envelope, which is a double membrane, and then it's continuous, which means it's connected to the ER, both the smooth and the rough. The other thing I like how this diagram shows is that the smooth and the rough ER are also continuous, which means they are not two separate organelles, but rather they're all connected through this membrane system. And the smooth can turn into rough, and the rough can turn into smooth, all based on whether or not these little dots are here. And these little dots represent ribosomes. Ribosomes on the ER make it rough. Okay, because it looks physically like it has little bumps, which it does, compared to the smooth ER with no ribosomes. Okay, so the smooth ER and the rough ER are this continuous network of membranes, and they're designated smooth or rough whether they have ribosomes or not. So smooth does not, rough does, and they can change. So if all these ribosomes over here came off, this area would become smooth. And so one way to think about this is if you had a couple of cups of Play-Doh. Okay? So hopefully you've had a chance in your life to play with Play-Doh. And Play-Doh could represent the membranes. And, oops, membranes. All, oh, I hate this. All membranes are phospholipid bilayers. All membranes. Okay. Just like all Play-Doh is Play-Doh, regardless of the color. Okay. The difference between all these membranes so they differ by their lipid content. And this is an important concept that we get into in cell biology. So there are things that are not just phospholipids, there's sphingolipids, and there's different backbones to these different lipids in the membrane. Cholesterol is important in membranes and found in different concentrations in different membranes. And the proteins in the membrane. Right? So in chapter 5, we'll talk about lots of proteins that are important in membranes, like transport proteins, carrier proteins, channel proteins. And then in chapter 8, we talk about receptors and signaling molecules that are on the plasma membrane. But what I want you to understand is that, like Play-Doh, membranes can interchange on these organelles in the endomembrane system, which means pretend you have a pile of purple Play-Doh and you have a pile of yellow Play-Doh. Well, you can pinch purple Play-Doh off and yellow Play-Doh off and you can mix it together, right? Or you could separate them and put them back on their pile. And that's exactly what's happening with membranes. They are, you can kind of see here, pinching off into something we call, well, not the orange one, here, we call vesicles. I'll talk about that in a minute. And you can see here, they're merging together, just like shoving orange and purple Play-Doh together. So membranes can be continuous. Right? And all membranes are made here in the smooth ER, because you have learned that the smooth ER is important for lipid synthesis, and membranes are made of lipids. Okay. Make sure I have covered everything. Okay, so that's the endomembrane system, this internal membrane system consisting of the nuclear envelope, the smooth and rough ER, the Golgi, lysosomes, and peroxisomes. Where the endomembrane system is most talked about is in protein synthesis. So we've talked about the macromolecule protein that's made up of hooking chains of amino acids together. And you may or may not remember that the information for proteins is stored in the DNA. So in chapter 10, we get into transcription, which is copying DNA to RNA, and translation, which is reading the mRNA and going to amino acids. So though you don't have to know the term translation yet, I want you to start being familiar with, this is the term for making proteins by reading the mRNA sequence and all translation happens on ribosomes. So ribosomes, yeah. So you've been memorizing all your parts of the cell and their function. Ribosomes are the site of protein synthesis. Some ribosomes attach to the ER, okay, and this makes it rough ER, but some ribosomes, let me change my, oops, see if green will show up. Some ribosomes here and here do not attach to the ER. So they do translation, they make proteins in the cytosol, okay, in the cytosol. Okay. So there are two sites of protein synthesis on the ER and in the cytosol. And so again, we're going to look at this endomembrane system, which is one of the ways proteins are made and distributed to where they need to go. Okay. This slide, I know it's a little blurry, but hopefully you can read it. Just to remind you of the players in the endomembrane system and what they do. So we talked about the smooth ER makes lipids. The rough ER makes proteins. And here's this term, it packages them. So whenever you hear the term package, think of these little membrane sacs called vesicles. Okay. And we have these different vesicles shown here and here on the left and right called transport vesicles. So their job is like a bus. They pinch off. So they were once this part of this membrane. They take this newly formed proteins, they pinch off and say, okay, we're going to take you over here. And likewise over here. They're transporting. Okay. There are also vesicles over here called secretory. You can read that. And the term secretion means to be released outside. So secretory vesicles take the contents from the ER and the Golgi, and then they spit them outside of the cell. So a really good example of this is the antibodies that some of your immune cells make to fight off an infection. They assemble these important proteins called antibodies, and then they secrete them out into the blood system so that the antibodies can go tag and help destroy foreign invaders. The Golgi, sorry, I wish there was a better way for me to do this. Golgi apparatus, a key word here is modifies. So the rough ER is going to synthesize and package, and it actually does a little modification, and we get into that in cell biology. But then the Golgi also modifies both lipids and proteins and sorts them and packages them. Whoops. And so you can see ER to Golgi to destination. So one of the destinations of proteins made in the endomembrane system is also lysosome. And you know that this is a digestive sac, so it helps break down old molecules and actually even old organelles. A little highlight that we will be talking about in chapter 5 is this process here, and this is called endocytosis. And again, you don't have to know this for chapter 4, but you do need to know it for chapter 5. And endocytosis is taking things from the outside and bringing them in. This pathway here is exocytosis, which means it's taking contents from the cell and making them exit or release them from the cell. what I want you to see is that this is just membranes making vesicles. And you can see again here a membrane and another vesicle merging together, right? Just like our Play-Doh. So you can pinch off and move it inside. You can pinch it and push the stuff outside. You can pinch the colors together inside because they're all membranes. They're all phospholipid bilayers. And so they can merge with each other and they can be pulled apart from each other. Okay, just another image. I like this one because it's kind of close up. And so you can see the rough ER continuous with the nuclear envelope. It has the little dots that are called ribosomes. And what's shown in green here is a protein. So this protein was made in the rough ER. It's transported by a vesicle to the Golgi. And what I really want you to see from this image is that the Golgi is a series of, or a stack, people like to say a stack, of membrane sacs. So it's actually different than the ER. The ER, everything is connected. Okay, and it's hard to see because it's supposed to be 3D. The Golgi is actually individual. So what this means is in the ER, the inside, which is called the lumen, is continuous, right? So you could put something here and just like a maze, it could follow all through the ER. That's quite different than the Golgi. These are, let's put individual membrane sacs. Okay, so their insides, their lumens, are not continuous. And that's because the Golgi sacs have different functions, right? So the cis face, which is closest to the rough ER, these sacs have different functions for protein modification and lipid modification and sorting than the trans sacs do. And trans is furthest from the ER and closest to the plasma membrane. And so you can see this little purple protein made in the ER goes and changes a little bit here. In the Golgi is moved to a transport vesicle, and this is actually, let me get rid of this. This is an example of a transmembrane protein, which we will talk about in Chapter 5, but I like to make sure you're getting exposure to the vocabulary because it crosses the membrane. Right? So it's trans across the membrane and it's a protein, and it did not get secreted. It just became part of the plasma membrane. So this might be a receptor molecule or something else for cell communication or cell signaling. Again, just another image. I think that if you can interpret images you haven't seen before, that means you really understand the concepts. So again, these red arrows are just showing protein synthesis pathways. Of the endomembrane system. And again, notice everything starts in the rough ER, goes through the Golgi, and then goes to its destination, which might be secretion, might be a vacuole, might be a lysosome, might be on the plasma membrane. It can actually come back and be part of the Golgi or part of the ER. And again, what we're looking at when we talk about the endomembrane system is this system for making proteins. And once again, remember that ribosomes can also make proteins just in the cytosol without the endomembrane system. So hopefully that helps a little bit. Keep reviewing the organelles and their functions, and we'll keep tying this together as to how they work together to make a cell function. All right, thanks.